Hello, good evening, doctors. Am I audible? Yes, hi, I'm Dr. Maimuna. Yes, today we are going to have the first live session of regular batch 2025. Yes, welcome to our class. Yes, today's class is about the placenta. So in this class, we are going to discuss about the anatomy of placenta, development of placenta and functions of placenta and the abnormalities of placenta. Okay, so about the introduction of the app, I'll be doing it later. First, we will go through the topic once. Okay, so my teaching would be more of a notepad session, like I'll be writing notes while parallelly I'll be teaching to you. Most of the times we will limit the teaching to the notes session only. We'll not go through the majority of lines in a PowerPoint presentation. I'll not read all the lines of the PowerPoint. Okay, I'll explain to you what is the most important things to read about, or what are the most important things you can learn from this topic, right? So just confirm me if you can see the screen or not. You can type in the chat box yes or no, and if the audio is clear or not. Yes, thank you. So yes, let us discuss about the anatomy of placenta. So the placenta contains two surfaces. The first surface is the fetal surface and the second surface is the maternal surface. Please stop me if you have any doubts at the end of the session, or you can just ping me. I'll reply to you in the chat. Or if you think if I'm going too fast or too slow, you can just let me know. Okay, feel free. Yeah, so we are having the two surfaces of placenta. This is the fetal surface and this is the maternal surface. Here, so many of you have seen the placenta live, so there is no point of discussion, but in the exam, you need to write what are the surfaces, what are cotyledons, what are lobes and everything. And basics are should be strong enough. Okay, so this is the fetal surface. The fetal surface, as you can see, this is shiny, this is smooth, and the umbilical cord also will be attached to the fetal surface. If any MBBS students have joined the discussion, umbilical cord will be attached to the fetal surface of the placenta. Okay, yes. Then comes the maternal surface. As you can see in the picture, this maternal surface is rough and it contains some lobular structures. These lobular structures are known as cotyledons, right? These lobular structures are known as cotyledons. So in the anatomy of placenta, you can write about, this is a disc-shaped placenta. Okay, this is a circular disc-shaped placenta and it is of around 15 to 20 centimeters at length. And you have to write the weight of the placenta. At term, the placenta weighs around 500 grams. At term, the placenta weighs around 500 grams, and the proportion of the birth weight to that of the baby and the placenta is equal to that of 1:6. Okay, so don't worry, I'll be giving you the notes as well for this, and I'll be telling you which topic is good and in which book. Okay, so the proportion of the weight of the baby and the placenta is around 1:6. So what happens at term? At term, the 4/5 of the placenta will be of the fetal origin. Previously, it would be of the maternal origin, but whenever the gestational age progresses, at term, there will be 4/5 of the placental fetal origin will be there. Okay. Okay. So, yes. Coming to the structure of the placenta. In the structure of the placenta, you have two plates, chorionic plate and the basal plate. See, what happens here is, this is the chorionic plate and this is the basal plate. So if you can see, at the chorionic plate, the umbilical cord will be attached. At the chorionic plate, the umbilical cord will be attached, and you can see the layers of the chorionic plate. So what are the layers of the chorionic plate? At first, it will be a primary mesenchymal layer, and then you will be having a cytotrophoblastic layer, and then you will be having a syncytiotrophoblastic layer. Okay, you will be having a syncytiotrophoblastic layer. So these are the layers of a chorionic plate. So actually this is not so useful in your academic sessions, but as these are basics, we need to learn about it. So this is the primary mesenchymal layer, this is the cytotrophoblast, and this is the syncytiotrophoblastic. This is easy if we go through the development of the placenta. So this will be discussed in this class only. So don't worry about the nomenclature. Okay. Then comes the basal plate. So here you have seen this is the basal plate. So this basal plate contains decidua basalis, okay, decidua basalis, Nitabukes layer, and the cytotrophoblastic shell also will be there, and the syncytiotrophoblast. Okay, if you can observe, you are having these two structures same in that of the chorionic and the basal plate. Okay, the chorionic plate and the basal plate will divide these two things. Okay, will share the cytotrophoblast and the syncytiotrophoblast. Okay, so this is about anatomy of the placenta. You can just write about the surfaces and plates. Surfaces are fetal surface and the maternal surface. Plates are chorionic plate and the basal plate. Average weight and average length and width of the placenta also you can write. Now, next, coming to the most important topic in obstetrics and gynec, when compared to that of the if you go with the questions or if you go in a practical life also, the development of placenta is very important. So I will be taking a long detailed discussion about the placenta, development of placenta. Okay, so if you don't want to forget any of the topics or any of the thing in the development of the placenta, you just need to write these 9 headings. Okay, what are these subheadings? The first step is the implantation. Next comes the formation of syncytio and cytotrophoblast. Then comes the formation of decidua. Then comes the formation of chorionic villi and lacuna, division of villi, placental barrier, cotyledons, and grading. So we will be discussing in detail about all these 9 stages. Okay, so in every stage you can write at least 1 or 2 simple points. That's it. No need of writing so much of lengthy things. Only the examiners or you will be remembering all the 9 things only, not all the nomenclature in the textbook. Okay, so first comes the implantation. So now I have drawn a diagram here. Let us see what is it. This is a morula. Okay, this is a morula. After fertilization, morula will be formed. Okay, after fertilization, morula will be there. At that point, the layer that is surrounding the morula, this is known as trophoblast. Okay, this is known as trophoblast. Then comes this is a inner cell mass. So here what happens after fertilization? The layer will be divided and it lines the outer epithelium that is known as the trophoblast, and the inner cell mass will be there where it is proliferating, where the mass will be proliferating. So what happens? This trophoblast. So what is meant by trophoblast? Trophoblast is very important. It has a capacity to attract anything, or whenever it comes into contact with any of the surface, immediately it wants to attach. So it has the property of attaching itself to any tissue. Okay, so that's why, to prevent the trophoblast attachment, this morula will be surrounded by zona pellucida. Okay, this morula will be surrounded by zona pellucida whenever it is traveling towards the uterus. Okay, so let us say if this is the fallopian tubes and if this is the uterus, and this morula will be there like this, but you have the trophoblast, so it can attach anywhere. That's why you have to have a protective covering against the morula, that is the zona pellucida. So whenever zona pellucida is there, this prevents the attachment of the morula to the tube or something. So this morula will be rotating, revolving, and it enters into the uterus. Okay, this picture we are showing in the uterus, right? If there is any problem in the zona pellucida, and if this zona pellucida breaks down prior to the entry into the uterus, then this zona pellucida will rupture and the morula will attach anywhere in this path, which leads to ectopic. This is the basic of ectopic. Whenever the zona pellucida is not good, if it is defective, then the morula can attach anywhere in its path, which leads to the ectopic. Okay, that is another story. So what happens here now? Now we need a proper fertilization. Now the morula has finished its task. It entered into the uterus. So what happens? The first step in the implantation is disappearance of zona pellucida. Zona pellucida should disappear. So this zona pellucida has disappeared. Then what happens? Slowly this morula enters into the uterine epithelium. This is known as the uterine epithelium. Okay, slowly it wants to erode or invade the uterine epithelium, and then the outer layer of the cells of the morula, they start growing abruptly and they start losing the cell boundaries. Okay, so these are the cell boundaries, right? So whenever they start proliferating, they will be having the cell boundaries. But what happens slowly, their cell boundaries will be lost. There will be only the syncytiotrophoblast, or there will be only the cytoplasm, and there are the floating nuclei. This is known as syncytiotrophoblast. Okay, that's where we got a differentiation of cytotrophoblast and you got a differentiation of syncytiotrophoblast. So syncytiotrophoblast will be having only the continuous sheet of cytoplasm containing many nuclei. That is known as syncytiotrophoblast. So this is all about the implantation, right? So when it occurs, this occurs at the 6th day of fertilization, whenever the morula has completed its journey towards the uterus and whenever the zona pellucida disappears, the invasion into the uterine epithelium occurs, right? Then comes so these two things are over, right? Implantation has been done and the formation of syncytial cytotrophoblast. At this juncture, at this point, this is known as blastocyst. Okay, from this point, we cannot call it a morula. Now it is known as a blastocyst. Okay, so 2 steps are over, easy. Next comes decidua. So what is decidua and why do you call it as a decidua? You can call it as, you can say it, no, why ma'am, why are you calling it as a decidua? You can say like endometrium. No, we cannot say endometrium, because in this decidua there will be so much action of progesterone at this point of time, where you will be having implantation and pregnancy is going to take place. So you'll be having so many reactions in the uterine endometrium, that is known as decidual reaction. At that point of time, the uterine endometrium is known as decidua. Okay, so what happens here is, whenever the implantation is done, the secretory phase features will be remained. So the secretory phase features will be remained. So uterus, menstrual cycle, as you know, you will be having proliferative phase and the secretory phase. But here what happens, whenever there is a pregnancy, the secretory phase will be remaining and we need to maintain that secretory phase. So what happens here, the stromal cells will be enlarging and then they will be leading to the formation of the decidua. In decidua, you are having 3 things. I will be giving you a notes as well, but if you are a person who have a habit of writing running notes, you can happily write it, no problem. So now coming to the decidua, you have 3 things: decidua basalis, decidua capsularis, and decidua parietalis. Don't worry, these terms are so much difficult, but if you see the diagram, you can just happily remember all these 3 things: decidua basalis, capsularis, parietalis. Now coming to the diagram, you can see, yes, this is decidua basalis. Okay, I am going to highlight this. This is decidua basalis. So what is decidua basalis? It is a portion of decidua where the placenta is to be formed. So now you can see, yes, so the placenta has been formed, right? So deciduo basalis is the portion where the placenta is forming. That is deciduo basalis. Easy. Next comes the deciduo capsularis. So this is deciduo capsularis. See, it is what it is doing. What it is doing, it is just capsuling. Okay, it is just separating the embryo from the uterine lumen. Okay, so this is the uterine lumen, you can see. Okay, this is the uterine cavity, you can see. So this deciduo capsularis is separating the embryo from the uterine cavity. It acts as a capsule, it acts as a shell over the embryo. That is deciduo capsularis. Next comes the deciduo paritalis, where the deciduo paritalis is lying here. So yes, so what is deciduo paritalis doing? The name itself shows paritalis, that is periphery, outside. So what it is doing? It is lining the rest of the uterine cavity. Okay, it is lining the rest of the uterine cavity. That is the deciduo paritalis. That's it. Only three things: basalis, capsularis, paritalis. Now you can say easily, no? So what is deciduo? The picture should come into your mind. Again, I can show you another picture for your easy remembrance. This is a little clumsy. You can see this schematic diagram. This is the embryo. Okay. So anyone, you can volunteer and you can say, so the deciduo which is separating the embryo from the uterine cavity, this is deciduo capsularis. Okay. It acts as a shell. Now comes the deciduo paritalis. Okay. It is just lining the other uterine endometrium. This is the deciduo basalis, from where you are having the placenta. Okay, done. Now you are having these three things are completed. Now comes the fourth stage, that is formation of the chorionic villi. This is one of the most important stage of development of placenta, where you need to pay attention here. So what is a villi and why you are going to discuss this? Because all the placental circulation, either it could be a fetoplacental circulation, or it could be a feto-maternal circulation, or it could be a maternal placental circulation. All these things will be through the villi only. In this, you are having three things, that is primary villi, secondary villi, and tertiary villi. So now you can see how the villi is formed. Okay. So let me show you with a diagram. Here, this is the extra embryonic mesoderm. You will see all the topics or all the terminology which we have discussed prior. Now you can see here. Okay, this is the extra embryonic mesoderm. Extra embryonic mesoderm. Now here you can see these are the blood vessels that are running through the mesoderm. So mesoderm is famous for vessels, nerves, tissues, everything. Okay, so mesoderm, the blood vessel is running here. Then what about primary villi? In primary villi, what happens is you will be having a layer of cytotrophoblast. Okay, a layer of cytotrophoblast will be protruding. A small layer of cytotrophoblast will be protruding, which is covered by another layer of syncytiotrophoblast. Okay, which is covered by another layer of syncytiotrophoblast. Now you can see this is cytotrophoblast, this is syncytiotrophoblast, right? This simply, it is a primary villi. You can simply say it is a central core of cytotrophoblast overlapped or covered by a layer of syncytiotrophoblast. This is the primary villi. Now coming to the second thing, this is the, okay. Now coming to the secondary villi. This is the extra embryonic mesoderm. Fine, happy. This is the vessels. Okay, happy. Again you are having the cytotrophoblast, correct. Syncytiotrophoblast, correct. What is the difference? You will be having in the secondary villi, you will be having, there will be a protrusion of the extra embryonic mesoderm. Okay, protrusion of the extra embryonic mesoderm. Here what happens? This extra embryonic mesoderm also gets protruded. Only difference between this and this is another layer of extra embryonic mesoderm will be joining it. That itself is a secondary villi. So now you can say primary villi is having how many layers? 2 layers. Cytotrophoblast, syncytiotrophoblastic. Now secondary villi is having how many layers? 3 layers. That is extra embryonic mesoderm, cytotrophoblast and syncytiotrophoblastic. Easy. What next? Next comes the tertiary villi. Yes. So in the tertiary villi, again you are having an extra embryonic mesoderm. Correct. Again blood vessels. Correct. Again you will be having a, what is this? Cytotrophoblast. Correct. Syncytiotrophoblastic. Correct. So in the secondary villi, you have improved to extra embryonic mesoderm has also added. Sorry. In the cytotrophoblast, tertiary villi, extra embryonic mesoderm has also added. Okay. All this is same with that of secondary and tertiary villi. Correct. So what happens in the tertiary villi? In the tertiary villi, there will be a presence of blood capillaries also. So the capillaries will say, no, no, don't leave me alone. I'll also come. I'll also come with you. I will join you. Okay, that itself shows this is a tertiary villi and this is a very well developed villi and this is a mature villi where all the layers are in synchronization and they go hand in hand. So now we can easily say what is a tertiary villi? Syncytiotrophoblastic, cytotrophoblast, extra embryonic mesoderm layer and the blood vessels. Okay, fine. Remember these things? These things, no? So we have discussed it somewhere. Yes. In the initial few minutes of the class, I have told you a very big name, that is chorionic plate, which you are afraid of. Now you can see this chorionic plate is containing primary mesenchymal tissue. Okay. Primary mesenchymal tissue, cytotrophoblast, syncytiotrophoblastic. It means it is containing a tertiary villi. Right? Or see, here you have gone to the conclusion that is a stem villi or a tertiary villi. Now you can understand, no, what it is? Yes. So that itself is a chorionic plate. So no need to worry of so many terminology. Here you have cracked it. This is about the villi. Now you have completed the another topic, that is formation of villi. Now coming to the lacuna, what happens? So what is a lacuna? How it is formed? I am going it in detail. I am going it slow because development of placenta is a very important topic and it is the initial topic which you will be learning in your clinical scenario in your gynec topics also. Okay. Yes. Now coming to the formation of a lacuna. So how it is formed? So what happens here is, again you will be going, again you will be having this villi. Now I will be having the cut section of the villi. So what happens here? This is the extra embryonic mesoderm. Okay. And then you will be having, so cytotrophoblast, which color? Yes. So you will be having the cytotrophoblast. Yes. Then you will be having syncytiotrophoblastic is just like a plate. So syncytiotrophoblastic, it is only a sheet of cytoplasm. Okay, fine. Then you will be having the decidua. Fine. So what happens here? So whenever there is a, this syncytiotrophoblastic is growing rapidly, which leaves some small cavities in it. Okay, which leaves some small cavities in it. This is a cavity, this is a cavity, this is a cavity, this is a cavity. So it leaves some small cavities. Gradually what happens? Usually at first they are arranged irregularly, like here one, they are arranged irregularly. But gradually what happens? They increase in size. Slowly the syncytiotrophoblastic grows and slowly they increase in size, which leads to the opening up of the lacuna. Okay, they will just combine. Okay, they will just combine, leading to a larger lacuna. So what happens when this larger lacuna is there? Whenever the larger lacuna is there, slowly the blood vessels will be traveling and the blood will be gushing here and then blood will be going out. This itself is known as a materno-placental circulation, and then it later forms into a feto-placental circulation also. So the blood vessels will going into the lacuna and then they will be coming out. This is about the another topic, that is the formation of the lacuna. That is very simple, important, and it is simple. Villi is only the most important one. Now lacuna is also done. Next, coming to the another important thing, that is division of rami. Another important thing is the division of rami. That is the sixth step in the development of placenta. So what happens here actually? So let us draw a diagram here again. In the if development of placenta is given, it is very easy, because you'll write only 2 or 3 points, then you will be drawing so many diagrams, then the examiner will think, okay, yeah, she has read and come, right? Okay, so yes, this is the diagram what we have drawn here. This is the maternal surface and this is the fetal surface. Okay, you can see my screen, right? Okay, this is the maternal surface and this is the fetal surface. So what happens here? So villi that are first formed, so the villi which we have studied here, the villi which we have studied here, the villi which are formed, they are attached to the fetal surface, to the mesoderm, and then slowly they want to go to the maternal side. Okay, slowly they want to go to the maternal side. How they will go? They will not go like this straight. They will have so many branches, so many deviations. Okay, so they will not go straight, and they will be having some rami, that means branches similar to that of a tree. Just imagine a tree. Tree will not just reach the sky. It will divide into branches, 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 and then it will go to the topmost thing. So the villi which has started at the fetal surface, it wants to go to the maternal surface, but it divides into several branches. Okay, that is known as a anchoring villi. The villi which is attached to this is known as the anchoring villi. This is the anchoring villi. So each anchoring villus will contain a truncus core. Okay, the villi, the overall villi is known as the anchoring villi, and now this trunk is known as the truncus core. Okay, we'll draw it. Yes, so you are having this trunk. So the whole villi is known as anchoring villi. Now this will have branches. So each anchoring villi will consist of truncus core. Okay, you will be having a truncus core. Then this divides into branches. These are the branches. So you are having this as a rami core. These are the rami core. Then it also divides into a finer branches. It is also again dividing into finer branches. These are known as ramuli core. These are known as ramuli core. Okay, so first is the anchoring villi, then truncus core, ramuli core, rami core, and ramuli core. So these are the things you need to remember. So these ramuli are attached to the cytotrophoblastic shell that is near to that of the maternal surface. Okay, this is about the types of villi. You have another villi. What it is? It is only a free villi. It will not go to anything. It will start here, but it will not go. It will be just floating, or it is known as a free villi. These free villi, they grow into the intervillous space. This is known as intervillous space. So they just grow like this. They'll not go to the maternal surface. They'll just grow like this and they'll stop there. These are known as the free villi. Okay, so what happens here? Why these are going from the fetus to the mother? Because fetus, every time it demands anything from the mother. So every time it wants anything, this will be transmitted to the villi, and all the blood from the mother will be traveling through these villi, and everything exchange will be taking place in the intervillous space. Okay, exchange will be taking place in the intervillous space. Okay, so this is about the attachment or division of the villi. Okay, so this is so much of topic for today, but please listen, this is very important. So we have discussed about the villi, right? Now comes the another one. Another one is the placental barrier. So why we are going to discuss the placental barrier? Because if you are having a so much of big community, You are having so much of big community, like a gated community. Anyone can enter or anyone can go. No, you will have a big security here. You will have a barrier here. You will have a barricade here. That itself is known as a placental barrier. So you are telling fetus is a parasite. It wants everything from its mother. So it has created so much of villi and it is just ready to accept everything. But still, this placenta will not accept everything to give. So it will limit some things. That which shows that thing is known as a placental barrier. So what are the layers of the placental barrier and how you are going to deal with it? So till now we have discussed the development of placenta and how it is growing towards the mother, such things. So what happens here? We are discussing here like the blood will be going like this and coming like this, but the maternal blood and the fetal blood will not mix. Okay, this is so much important because you will be having the you will be having against immunity like foreign body reactions or something, no? So these are not possible because of the placental barrier. So this is not an anatomical barrier. You cannot see a big barricade or a gate at the placenta. This is a physiological barrier where you will be having small layers or a thin layers will be there. And here you can see this is not a perfect barrier as such, because sometimes the maternal blood cells will be found in the fetus and the fetal blood cells also will be found in the maternal circulation. So that's why we are going to see this. And coming to the composition, so what is the placental barrier and how we are going to deal with it? So in the early pregnancy and in the late pregnancy, how you are going to see this? So in the early pregnancy, what happens is there will be syncytiotrophoblast and cytotrophoblast. In the early pregnancy, you will be having syncytiotrophoblast and cytotrophoblast. But later, what happens? In it will be having the basement membrane, stromal tissue, and endothelium also. I will be showing a picture also. Don't worry. And in the near term, what happens? You will be having a cytotrophoblast will be very less. Cells will be very less. If I draw a diagram, then you will understand it easily. So in the early pregnancy, what happens? Just draw a diagram. Okay, these are the cells. Okay, this is the stroma. Now this is coming to the connective tissue. This is the connective tissue. And again, you will be having a basement membrane. Okay. Then you will be having the cytotrophoblast. Then you will be having a syncytiotrophoblast. Syncytiotrophoblast we have thought, no, this is only just a layer of cytoplasm. Syncytiotrophoblast. Now here you will be having the maternal blood. Okay, maternal blood. So what happens here? In the early pregnancy, you will be having all these things. Okay, you will be having connective tissue, you will be having stroma, you will be having basement membrane, cytotrophoblast, syncytiotrophoblast, maternal blood vessels. Everything will be there in the early pregnancy. But what happens whenever you go like if it is going to the term, in the near term, this syncytiotrophoblast will be very less. Thin syncytiotrophoblast will be there. So if we draw a cut section, you will remember it clearly. This is all a syncytiotrophoblast. Okay, this is all a syncytiotrophoblast. And here you can see these are the cytotrophoblast. This is a cut section. This is a cytotrophoblast. And these are the vacuoles. Somewhere you will be having for the circulation, these are the vacuoles. The vacuoles will be small, and you will be having mesodermal core and all other such things will be there. And this is a capillary. Okay, just this diagram is a schematic diagram just to make you remember that in early pregnancy what happens and in the late pregnancy what happens. Okay, So this is a syncytial trophoblast, and this is cytotrophoblast. So in the early pregnancy, you are seeing a very thick syncytium, very thick cytotrophoblast, okay, and very small vacuoles. But what happens if it is near term? If it is a near term, you can see syncytial trophoblast becomes sparse. Only like a thin layer you will be having. You will be having a very thin syncytial trophoblast, and the vacuole will become so much big. Okay, vacuole will become so much big because the placental circulation takes place there only. That's why the vacuole becomes big, right? And now here you can see sparse cytotrophoblast. Only at very less points you can see the cytotrophoblast. Yes. So this is the placental barrier. What happens here? You will be having a thick syncytial trophoblast and you will be having a thin syncytial trophoblast. Why? Why this is important? Thin syncytial trophoblast, you will be having a very good gas exchange. Very good gas exchange to the baby. Very good gas exchange to the baby. So what are the functions of a what are the functions of a placental barrier? Coming to the functions of a placental barrier, where you can see it will be having a interchange of the material. Okay, you will be having the interchange of the oxygen, nutrition, and all the other waste products. And then the total area of the membrane would be around 4 to 14 square meters will be there. So what happens towards the end? Slowly there will be fibrinoid deposits here and there. There will be fibrinoid deposits here and there, and whenever the term progresses and if it enters into the post-dated pregnancy, then what happens? The fibrinoid deposits will be more, calcification will be more, and it leads to the decreased efficacy of that membrane, and gas exchange will be less. That leads to sudden intrauterine deaths or meconium stained lichens. So whenever you go to the extreme depth of gynecology, you will see post-dated pregnancies, you will have more meconium stained lichens. Post-dated pregnancy, you will have more stillbirths, you will have more IUDs. Why? Again, you have to come to the basic anatomy, that is fibrinoid deposits will be there in the placental barrier, which decreases the gas exchange. Okay. So in the syncytial trophoblast, you are having two things, that is a thick zone and a thin zone. The thin zone is for the gas exchange, and a thick zone, this is known as a beta zone, this is known as an alpha zone. These are less important, so I'm just giving you a crisp. The thick zone is a beta zone. It will be having patches, and this is useful for the hormone synthesis, right? This is for the hormone synthesis. So this is about the placental barrier. Yes, thankfully we have completed the placental barrier also. That is another topic of the development of placenta, right? Next comes the formation of cotyledons. So yes, what we are going to do, formation of the cotyledons. Now what happens? The placenta has grown into a very full mature placenta, right? It has formed the villi, it has formed the decidua, then now it is a fully mature placenta. So what it is going to do? It will be divided into lobes. It will be subdivided into lobes. Slowly there will be formation of the lobes, and slowly there will be formation of the cotyledons. So whenever there is a lobe plus and anchoring villi, and if the branches are also there, all these comes under fetal cotyledons. The fetal cotyledon is formed by a lobe, anchoring villi, and branches. That is known as the fetal cotyledon. Usually fetal cotyledons will be more, like around 60 to 100, but maternal cotyledons are less. It will be having only 15 to 20 lobes in the placenta. You will be having 15 to 20 lobes in the placenta. Okay. So this is about the formation of cotyledons, nothing much in it. Now comes the another important one, that is a grading. So how do you grade the placenta? Okay, grading. How do you grade the development of placenta? So whenever the cotyledons are there, you will be having the granums stage of grading. This grading is known as granums grading. So what happens in the granums grading? In the grade 0, it will be a homogeneous placenta. Okay, it will be a homogeneous placenta, only smooth chorionic plate will be there, and only small cells will be there. Okay, this is the grade 0. In grade 1, what happens? The echogenic densities will be randomly dispensed, and usually it occurs in the, these are the echogenic densities, and usually they settle at the base. They settle at the base. It is the grade 1. Now comes the grade 2. In grade 2, what happens? Slowly indentation in the chorionic plate will be there. Slowly there will be an indentation, but it will not touch the basal layer. Okay, it will not touch the basal layer. Only indentations will be there, and slowly the echogenicities also will be increasing. The echogenicities also will be increasing. Now comes the grade 3. In grade 3, what happens? Fully grown cotyledons will be there. Fully grown cotyledons will be there, and more marked indentations will be there, and the echogenic densities also will be collaborated, and it will be like a vacuole, or it will be like an echogenic area in the center of the cotyledon. Echogenic area in the center of the cotyledon. So this is about the Granum's grading, or the grading of the placenta. Only simple steps. Like if you have no time writing it, you can just simply write the grading of the placenta, that is 0, 1, 2, 3. Okay. Okay. So this shows the development of the placenta. Now the placenta has fully grown. The cotyledons have grown, and the villi have grown, and the interplacental circulation is going on. Okay. Now the placenta says, yes, I have grown up. Now what I am going to do? Okay, now what I want to do? So now the placenta says, yes, what are the functions of the placenta? So if you are thinking of a placenta, then you can immediately say the functions of the placenta. You can have in the, yes, it will be an endocrine function. It will be a nutritive function in which there will be a gas exchange. Okay, it will be a nutritive function where there will be a gas exchange, and then you will be having a hormonal function. Okay, you will be having a hormonal function, and then as we have discussed, you will be having a barrier function, right? So these are the functions of the placenta. Here you can see. Here you can see, yes, this is the functions of the placenta. In the functions of the placenta, you can see, like the placenta will be transferring all the nutrients, oxygen, gas, exchange, everything will be there. And then if you are talking about the endocrine function, the placenta will be releasing HCG, inhibin, relaxin, everything will be released by the placenta. Then the barrier function, we have discussed placental barrier, and then the immunological function to prevent the foreign body reaction to the fetus. So the functions of the placenta, if you want in detail, you can read it in Datta. It is given accurately in Datta also. Now, functions is also completed. Yes, good that we have completed 3 things, that is anatomy, development, and then functions. Then comes the abnormalities of placenta. Okay, so what we are going to study in the abnormalities of placenta, where the placenta will be implanted, that itself will show if any abnormality or not. So we can see the abnormalities of placenta. It is given very good in Inderbeer Singh. That is Inderbeer Singh, IB Singh, page number 71. That is Inderbeer Singh embryology. Okay, Inderbeer Singh embryology, where you can see the abnormalities of placenta. Don't worry, every topic you cannot find full-fledgedly in one scenario. You can find it in very different books, so you have to just correlate all the things, and we compiled it in our notes, so that if you are a final year, don't just rush to all the books and don't try to compile the things. We have made the things easy for you. We have created all the thing in our notes, and in our class also we have collaborated so many things in one place. Okay, so you can just go through the class, and then you can go through the notes. That itself is enough. So now, what are the abnormalities of placenta? Whenever there is a implantation of the placenta is not correct, then it will be abnormal. So this is usually the normal site, that is at the fundus, that is normal site. But if it is not implanting here, if it is implanting just below the fundus, or if it is implanting to the lateral side, or if it is implanting nearer to the os, then it is known as the placenta previa. Now, what again, it is a interstitial. If the placenta is implanted here, that it is known as interstitial tubal implantation. If the placenta is implanted in the tube, then it is known as a tubal implantation, which might lead to ectopic. It means if the placenta is implanted here, what it means? The embryo is also implanted here. No. What is the function of placenta? To create food for the embryo. So wherever the embryo is there, placenta will be there. No. That is the reason you will be having a tubal ectopic here if the placenta is implanted here. You will have a abdominal implantation, or you will be having a ovarian implantation. These are the abnormal sites of placenta. Now you can see the anomalies of placenta. So what are the anomalies of placenta? You can see. So in this first one, you will be having a bidiscoidal. You will be having 2 discs of placenta. Okay. The second thing, you will be having lobed placenta. Lobes will be there. And in the third thing, you will not have any structure to the placenta. All the capillaries are diffuse. All the capillaries are diffuse. That's it. That is known as a diffuse placenta. And in the D, that is suction turate placenta. Suction turate placenta is one lobe of the placenta will be implanted away from the normal site. So the placenta will be implanted at the same site, but one lobe will be separate. That is a suction turate lobe placenta. And in some places, this is a fenestrated placenta. You will be having a hole in the placenta. That is a fenestrated. And the final one, this is a circumvallate placenta. Okay, where there will be a fold of decidua, and the placenta will be just down, and the umbilical cord also will be attached to the margin. That is a circumvallate placenta. Okay, these are the abnormalities of placenta. If you want to go in detail, you can go through the Inder Singh page number 71 also. And the pictures are also taken from these only. Now coming to the placenta previa types. Okay, so what happens in the placenta previa, and if it is implanted inside the uterus, just down below the fundus, then it is known as the placenta previa. You are having grades of the degrees of the placenta previa. In the first degree, you can see, yes, this is the uterus, and here this is the placenta. This is the normal site. This is the first degree of placenta, where it is extending into the lower uterine segment. Where is lower uterine segment? This is lower uterine segment. It is extending into the lower uterine segment, but it does not reach the os. It does not reach the os. It extends into the lower uterine segment. Now comes the second degree, where the margin reaches the os, but it does not cover the os. Okay, it does not cover the os. In the second degree, you will be hearing a terminology margin. In the second degree, you will be hearing the terminology margin. Just a second. But in the second degree, you will be hearing margin. But in the third one, you will be having the edge. So edge of the placenta will be there. Okay, edge of the placenta will be there. Can you see my screen now?
No, ma'am.
Okay, just a second. Okay, so coming to the grades of placenta previa, the first one will be like it will be just entering into the os, and it will not cover the os. And the second thing, the margin will be reaching the os. Now comes the third thing, the edge of the placenta will be covering the os. And in the fourth degree, what happens? The placenta will be completely covering the os. Don't worry if the screen is not sharing, because we have come to the end of the class, and this is about the placenta previa. And yes, now I think you can see the screen. Yeah. Okay. So this is about the placenta previa degrees. In the first degree, it is extended into the lower uterine segment, but it does not reach the os. But in the second degree, what happens? The margin reaches the os, but it will not cover the os. Here in the diagram, you can see clearly, it has not covered the os. Okay, it has not covered the os. Now comes the third degree. The edge of the placenta has covered the os. Okay, edge of the placenta has covered the os. But here one thing is there. If you can see a third degree placenta, and if the edge is covering the os, but whenever the patient enters into labor, after the cervical dilatation, this placenta will slowly go up. It will not hinder the birth of the baby. It will not hinder the birth of the baby. But what happens in the fourth degree? The placenta has completely covered the os. So what are the things which you can see in the placenta previa? Or you will be having immense bleeding, immense postpartum hemorrhage or antepartum hemorrhage also you could see. Okay, that is the reason why you are worried about the grades of the placenta, or why you are so much worried about the placenta is because it will be having a huge blood supply, and it can immediately go into labor also. Okay, now coming to the abnormalities of umbilical cord insertion. Why we are discussing here is because we have discussed so much about placenta. Umbilical cord is its friend. It always attaches to the placenta. So just only the important points in the umbilical cord is, usually this is the normal attachment of the umbilical cord. It attaches here at the chorionic plate. But what happens here in this, it is attaching at the margin, that is known as a marginal insertion. And if it is attaching as a branches, that is a bifurcate, that is a furcate insertion. And then if it is attaching from the side, okay, that is known as the velamentous insertion, right? So this is about the umbilical cord insertion. So now we have covered about the, what are the things we have covered in the today's class. Today's class we have covered about anatomy, development, functions and abnormalities of placenta. Okay, so in the anatomy, yes, we are well versed now. We can immediately say 10 to 20 points about placenta development. So if anybody asks about development, you can immediately say these 9 points. Okay, so whenever a question comes with the anatomy of placenta, development of placenta, functions of placenta, you can just write these things. Okay, don't worry, we will be preparing we'll be having a notes for you, and we will share it with you, right? So this is all about the anatomy and development of placenta. See you in the next class. Thank you for joining. So like next week we will be having another class for the regular course people for MS and DNB, where we will be discussing about placenta previa, abruptio placenta, placenta accreta spectrum. Okay, so whenever you will have a strong basic knowledge of such things, the other things like the depth things and further things will be easier. That is only a cream on bread. Okay, so the making of a base is important. So if you have learned so much about the basics of placenta, you can immediately say, okay, this defect is there, then this is the problem. This defect is there, then this is the problem. You can immediately say that, right? So basics are important. Thank you for joining, and thank you for your patient listening. So once again, I would like to
Ma'am,
say,
where we
please
can we
download the
have
app. You
the
can have some free videos.
recorded
Sorry,
video?
if anyone is
Where we
asking
can see
something.
the recorded video?
Yes, we will be sharing the recording video with you in the YouTube and in the app also. So you can join the YouTube, or you can download the app, in which you will be having a free video section where you can see this video, right?
Thank you, ma'am.
Okay, right. Thank you so much for joining and patient listening. Thank you and happy learning.